From the American Neurological Association, this is ANA Investigates. I'm Adeline Goss. The role of genetics in both pediatric and adult epilepsies is expanding. Now, making a genetic diagnosis in epilepsy isn't just important for family planning and prognostication. These diagnoses can also help improve patients' care starting at the moment of diagnosis. Our guest today, Ingrid Scheffer, is a leader in gene discovery in epilepsy. Dr. Scheffer is a Laureate professor of pediatric neurology at the University of Melbourne, and she directs the Children's Epilepsy program at Austin Health. She says while there's still a long way to go before precision medicine is available for the hundreds of known genetic epilepsies, her patients are already benefiting from genetic testing in many ways. Dr. Scheffer was interviewed by ANA Investigates producer and epileptologist Dr. Rohit Das of UT Southwestern Medical Center. Here's Rohit.
Welcome, Ingrid. We are so happy to have you here speaking on ANA Investigates.
Lovely to be with you, Rohit.
Ingrid, let's start with the big picture. How has genetics changed our understanding of the epilepsies?
Well, really, genetics has revolutionized our view of the epilepsies by giving us far greater insights into the neurobiology of this severe group of disorders. And what we're understanding now is where genetics offers us the answer to why a patient has epilepsy. And in some of those patients, we're talking about monogenic causes, such as the most severe group of epilepsies, the developmental and epileptic encephalopathies, where lots of both children and adults have a cause that can be due to a dominant de novo pathogenic variant, or perhaps recessive or X-linked. On the other hand, though, we're finally beginning to get some insights into complex genetics, which is where more than one gene is contributing. Often we're thinking about a polygenic background. And here we're talking about common forms of epilepsy, such as the idiopathic generalized epilepsies, and even in some cases, the sporadic focal epilepsies.
What do we know about how epilepsies can be inherited?
Well, this is really fascinating and occurs at multiple different levels. So the obvious thing is where you've got a large family with several generations of affected individuals. And then old-fashioned clinical genetics tells us that this could be inherited in an autosomal dominant way. Now, in those large families, which are rare, it's obvious it's genetic. But what we found in the last 30 years now, it's very exciting because we now know that genetics contributes to many forms of epilepsy. So it may be that can be not inherited. If it's a new mutation in the child, then the child hasn't inherited it from mother or father. And that's very important because often families may have a feeling of guilt that they've given their child their epilepsy, and yet it's a new mutation in the patient, so they're not inherited. So the first thing is it inherited? Often not. A genetic cause does not equate to an inherited cause. They're two different things. But one of the most exciting facets of the field is that we're now finding quite a lot of pathogenic variants and mosaic. So what I mean by mosaic is you have two populations of cells. You'll have your mutant population of cells with a pathogenic variant, and the rest of the cells are wild type or normal. Now, if a person is mosaic for a pathogenic variant, then that variant must have arisen newly in that person. They can't have inherited it. And then they may not be at such great risk of having an affected child, depending on where that mosaicism is. So, for example, we're finding quite a lot of malformations of the brain are due to mosaic pathogenic variants in that part of brain, but that pathogenic variant may not be present in other tissues. So it really needs quite a sophisticated understanding of the different modes of inheritance to be able to understand whether a specific form of epilepsy is inherited, and therefore to be able to give accurate reproductive risks.
Thank you so much for talking about that. When should we pursue genetic testing?
It is really important to pursue genetic testing in all the patients that you see, be they adult or child. And particularly, let's start with the severe end, the developmental and epileptic encephalopathies, which which is probably around 10% of the patients that we see with epilepsy, all of those patients should have a copy number variant study to start with, which is a chromosomal microarray, because this will look for abnormal duplications or deletions that may be either wholly causative or contributing to the etiology in the patient. And staying with that very severe group, we should also be now pursuing exome or even ideally genome sequencing in this group, because you are likely to find now 50% of the patients we can solve in that we find a causative pathogenic variant, a monogenic causative pathogenic variant. Now let's turn to the more common group of our patients, our otherwise very normal patients with idiopathic generalized epilepsies or focal epilepsies. And most of these patients require a chromosomal microarray to look for a pathogenic copy number variant. Mostly you don't find them, but in about 3% of the common idiopathic generalized epilepsy patients, you will find a recurrent copy number variant, which is a contributing cause, if you will, to that person's epilepsy. And you can say, well, what difference is it going to make if I find that? Where it can make a difference is they may have a risk to their offspring, and the risk may be greater than just idiopathic generalized epilepsy, as some of these copy number variants increase your risk of intellectual disability, autism spectrum disorder, schizophrenia. So, you know, if you find them, then the person needs to have formal genetic counselling. In terms of the common epilepsies, and we're sticking with the idiopathic generalized epilepsies and the focal epilepsies, is there a place for genome sequencing or exome sequencing? Well, there are rare monogenic causes of those disorders, and they can have other consequences and also have reproductive counselling risks. So I think it is worth doing it, but obviously this isn't accessible to many of those patients. The other area where the landscape is absolutely changing for these common epilepsies is the testing called a polygenic risk score. Now, where this is fascinating, so a polygenic risk score derives from a genome-wide association study. And just to explain that in a few words, it's where you take ideally tens of thousands of patients with a disease X and even more thousands of control patients. And you ask the question, where do those tens of thousands of patients with disease X share the same area or SNP, single nucleotide polymorphism on chromosomes? And then you build this Manhattan plot where you can actually see where they share more of the same areas. And from there, you can then derive a polygenic risk score where you say this patient is at more risk of having this disease because they have a higher polygenic risk score. So whilst that hasn't reached the clinic yet for testing, I don't think it's far off. And I think it will reframe how you use genetic testing in the clinic.
Leading on from there, what are the different genetic tests available? And could you describe the different methods of testing?
Right. Well, there are many different levels to genetic testing. Just to start with the first sort of screening test, this chromosomal microarray. You can do that now very easily on buccal testing or salivary testing as well as blood. So that is useful if you have an intellectually disabled patient that it's hard to get, who is hard to get blood from. You can then just get it from some saliva. And that's a test that you send off to the laboratory and they'll come back with whether there's any significant copy number variant testing. When you're actually looking for a single gene abnormality, such as a missense pathogenic variant or a truncation pathogenic variant or a small deletion within the gene, then you often need to have exome or genome sequencing. Now, until now, we've been largely using gene panel sequencing, but exome or genome sequencing takes us to the next level. And I think that is changing, that we will be moving from a panel to exome sequencing. And these are all different forms of next generation sequencing. And with an exome, that means you're sequencing all the base pairs in about 1 to 2% of the genome. and that's in the exons. And remembering that the exons are the parts of the genes that encode the protein. So we know that many of the pathogenic variants lie within the exons. So these are the highest yield. And really the holy grail now is genome sequencing, and that is where you sequence every part of the person's genome. But we still find that a bit harder to interpret. And that's happening, certainly at a research level. We're doing a lot of genome sequencing. And I know some laboratories are offering genome sequencing. When the labs do the exome or genome sequencing, they will send you a report with the known epilepsy genes that they've looked at particularly, and they'll offer their thoughts about whether the variant they've found is definitely pathogenic or not clearly so. But one of the issues is that new genes are being discovered all the time. So if your exome or genome sequencing is negative today, in 6 months or a year or in 3 years, they may discover that there's a new gene in which your patient has a known variant, and that's the cause. So it has to be iterative. You have to keep looking at these results and asking the labs to reconsider them every few years.
Ingrid, how does finding a genetic mutation help with prognostication?
Finding a genetic mutation actually changes your management of the patient from the day you get it. People seem to think, oh, it's all in the future. We're looking at precision medicines, and that is certainly the exciting future. But I find that getting the result changes my management on that day. And just to give you an example, last year I saw 2 different unrelated babies. They'd both had infantile onset seizures. They'd both been put on levetiracetam. Neither was doing that well. On the day that I got the SCN8A pathogenic variant in one baby, which was actually inherited, I changed her, and she was actually going off developmentally. I changed her to carbamazepine and stopped the levetiracetam over a few weeks. She has been seizure-free since and has had made normal developmental progress. That's just one example. The other example was another baby, a little boy, that was on levetiracetam, actually also had a paternal family history. The father had seizures from 3 to 7 years and was very normal. And to my surprise, this little boy returned an SCN1A pathogenic variant, and he'd been having sort of 15-minute seizures that were focal in nature. And so that was a game changer, because suddenly I realized this little boy probably had Dravet syndrome. And you can say, well, how could dad have had it and have grown out of his epilepsy? Well, there was one answer to that, and that was that dad was likely to be mosaic. So dad didn't have the mutation in every cell in dad's body, and that's why dad was normal and grew out of it. And indeed, we went on to study dad and showed that was the case. Now, how that changed the management of this little boy was on the day that we got his SCN1A result. Levetiracetam isn't very useful for Dravet syndrome, and we swapped him from levetiracetam to valproate. And these days, we're finding quite a lot of neurologists are putting these babies onto carbamazepine, which of course is really bad for Dravet syndrome. And so it actually makes the seizures more frequent. So the sooner you stop the carbamazepine or the oxcarbazepine, you can improve seizure control. So from the day I get the result, it changes management. The second thing to say is that a genetic test will also change you from a syndromic diagnosis. Well, it will add to it in a complementary way. So you'll have both a syndromic diagnosis and an etiological diagnosis. With an etiological diagnosis, and again, I'll just go back to Dravet syndrome, you can actually start to look for the associated comorbidities that we see with these diseases. So in an adult with Dravet syndrome, you'd be looking for the crouch gait. In an adult with PCDH19 clustering epilepsy and a woman with that, you'd be worried about psychosis. So actually understanding the genetic etiology informs multi-morbidity understanding and also informs longevity. So we know that some of these genetic epilepsies have a risk of SUDEP, but others do not seem to. So this can actually offer a lot of reassurance, or for families, if they have a genetic disease that is not associated with SUDEP, sudden unexpected death in epilepsy, Or alternatively, it may make the family more vigilant, and maybe they'll put a camera in their room or whatever to try and look out for that risk of SUDEP. So it helps with treatment from the day you get the result. It helps with understanding the comorbidities, and it also helps with prognostication. And often the mothers of these patients, and fathers, but the mothers more so, have really been worried that it was this glass of wine I had when I was pregnant that gave my child this terrible disease. And when you find the genetic abnormality, they can realize it was nothing to do with that glass of wine. It was just simply bad genetic luck. And we all have that risk when we have a pregnancy.
Are there specific treatments that open up for patients once we identify a genetic mutation?
Oh, that's a super important question, Rohit. And I think this we're really coming into the era of precision medicine. And precision medicine means that you have a treatment that is targeted to the underlying genetic etiology. In some ways, one can think of precision medicines at several levels. The first level is, as I was just explaining before, from the day you get a result, say of SCN1A in a patient with Dravet syndrome, and you stop carbamazepine or oxcarbazepine, you can see improvements. And there's a really nice paper from 2011 from Katerina and colleagues. And in that, we showed that if you stopped carbamazepine in a 60-year-old with Dravet syndrome, they could improve cognitively. So that's the first thing. The second thing is with precision medicine. There are some beautiful examples of where we can already use this. And one of those is Batten's disease, or neuronal steroid lipofuscinosis. And here I take the example of CLN2 disease, which has been fatal until the development of a precision medicine in the form of enzyme replacement therapy. And in this particular precision therapy, the patient has a recessive disease and is missing their enzyme TPP1. And via intracerebroventricular pathway, these children get 2 weekly infusions of replacement enzyme therapy. So this isn't actually gene therapy, it's just giving the patient back what they're missing. In terms of gene therapy for the epilepsies, there's a huge amount of excitement and interest in this. There are 2 gene therapy trials that are underway or about to be underway for Dravet syndrome. One is an antisense oligonucleotide, which is being led by Stoke Therapeutics. The trials have started in the US and Europe, and I know they're looking at dose finding and safety, which are the first steps with gene therapy. And the other one is being launched by Encoded, and that's a different type of gene therapy where you change the regulation of the SCN1A gene in the good copy. So you ignore the bad copy that doesn't work, but you try and upregulate the good copy. So it's very much watch this space, and I think it will be transformed in the next 5 to 10 years as we determine gene therapy. But remembering that, as I said at the beginning, we have 800 genes, so that's 800 gene therapies. So we've got a long way to go.
Thank you so much, Ingrid, for speaking to us about genetics and epilepsy. This was a fascinating talk, and thank you again.
My pleasure.
That was Dr. Ingrid Scheffer, Laureate Professor of Pediatric Neurology at the University of Melbourne, interviewed by Dr. Rohit Das, Associate Professor of Neurology at UT Southwestern Medical Center. That's it for ANA Investigates. I'm Adeline Goss. You can still visit myana.org to register for the ANA's annual meeting from October 22nd to the 25th in Chicago. Our next episode of the podcast will be in November. See you then.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association, and an ANA Education Innovation Subcommittee production. Thanks for joining us. To learn more about today's program and to find out about the benefits available to ANA members, including access to an array of educational programs and resources, please visit myana.org. Dr. Adeline Goss is our host and the executive producer of the show. Episode producers are members of the ANA Education Innovation Subcommittee, chaired by Drs. Megan Ritchie and Romer Giaccaden. This is your logistics coordinator, Jen Hurley. Copyright, the American Neurological Association.